Metagenomics is the study of genetic material recovered directly from environmental samples, while microbiome refers to the collection of microorganisms that inhabit a particular environment. The connection between metagenomics and microbiome lies in the fact that metagenomics can be used to study the composition and function of microbiomes. By analyzing the genetic material present in environmental samples, researchers can identify the microorganisms present in a particular microbiome and determine their roles in that ecosystem. This information can then be used to better understand how microbiomes function and how they are affected by different environmental factors. By sequencing the DNA or RNA extracted from a sample, researchers can identify the different species of microorganisms present, as well as the genes and metabolic pathways they use to interact with each other and their host. Applications of metagenomics in microbiome research. By analyzing the genetic material present in fecal samples, researchers can identify the microorganisms present in the gut microbiome and determine their roles in digestion, metabolism, and immunity. This information can then be used to develop new therapies for diseases such as inflammatory bowel disease and obesity. Challenges in metagenomics and microbiome research. Despite its many advantages, metagenomics also presents several challenges in microbiome research. One of the biggest challenges is the sheer complexity of microbiomes, which can contain thousands of different microorganisms with varying functions. Another challenge is the difficulty in accurately identifying and characterizing the genetic material present in environmental samples. This requires the use of advanced sequencing technologies and bioinformatics tools, which can be time-consuming and expensive. Ethical considerations in metagenomics and microbiome research. As with any emerging field, metagenomics and microbiome research raise important ethical considerations. One concern is the potential for misuse of personal microbiome data, which could be used for discriminatory purposes or sold to third-party companies without consent. Another concern is the impact of microbiome research on vulnerable populations, such as indigenous communities, whose microbiomes may be threatened by environmental degradation or loss of traditional lifestyles. These concerns highlight the need for responsible and transparent practices in metagenomics and microbiome research. In conclusion, the connection between metagenomics and microbiome is a powerful tool for understanding the diversity and function of microbial communities in different environments. While there are many challenges and ethical considerations associated with this field, the potential benefits are significant, including the development of personalized medicine and novel therapies for diseases. As metagenomics and microbiome research continues to evolve, it will be important to balance scientific discovery with responsible and transparent practices that respect the privacy and dignity of all individuals and communities involved.